Hello and welcome to Bioaffairs. So today I'll be talking about the third part and the last part of virology, where I'll be discussing about its pathogenicity, its cultivation, diagnosis, treatment and prophylaxis. So with this we will be completing the viral chapter, overall viral chapter, because each virus is different, their actions are different, their mechanism of action, their infection cycle, everything is different, their genome. So let's start with the virology decoded part 3. So first we will be seeing the pathogenesis at cellular level, what happens when a virus infects you. So first of all is the incubation period. There are multiple viruses roaming around you, but among them the genetic characterizations are different. So what happens, the incubation period, first you have to know the incubation period. It is the period from where the virus enters into your cell or enters into your body and the first onset of symptoms. This period is called the incubation period. Now this incubation period depends on several factors, and from one virus to another it varies. So I am not going to particularly give you the detail of each virus's incubation period, but you need to know the term incubation period. What is incubation period? So I think you are clear that the incubation period is the time interval between the entry of the virus. So here it is entry of the virus where? To your body or your cell, and appearance of the first clinical manifestation, any clinical sign, that is fever, cough or any kind of rash for a particular virus, whatever it is. So this period is called the incubation period. That is very important. Now coming to the pathogenesis, there can be two types of pathogenesis. One is complete lysis of the cell, that is the cell death. Okay, the cell death is the ultimate possible thing that can happen by the viral infection, or a infection where no cell death takes place, or cell is as it is and they are increasing their number one by one. Okay. So inhibition of host cell DNA by herpes virus. I have given some examples of the viruses which are mainly responsible for lytic infection of their life cycle. That is herpes virus, which inhibit the host cell DNA. The mechanism of growth in their scenario is inhibition of host cell DNA synthesis, whereas in case of poliovirus, inhibition of cell protein synthesis. So what will happen if you inhibit the DNA synthesis or the protein synthesis? There will be no multiplication or any kind of basic work what is going on in your cell. Metabolism will be stopped. Now then fusion, syncytia formation. Syncytia is a term which is used for cellular fusion mechanism or cellular fusion phenomenon, you can say. So glycoproteins of some enveloped viruses or paramyxoviruses, herpes viruses, retroviruses, what happens? They induce the cellular fusion, means two cells get fused with each other and make a multinucleated giant cell, because one cell contains only one nucleus. So if multiple cells fuse within each other, what will happen? The nucleus will be also, the nucleus cannot be destroyed now. So as it is in that situation, a cell which is large enough in size and also contains multiple nucleuses. So it is a multinucleated giant cells called syncytia. Okay, remember this, multinucleated giant cells called syncytia. Now immune mediated lysis, what happens? Now due to some viral antigens, not all viruses, again I am telling you, due to some of the viral antigens, the host cell surface can lead to binding of antibodies. Means suppose a virus infects your cell, any tissue or cell in the body, what happens? Their antigens get attached to the cellular surface. Now that antigens attract antibodies. Okay, their antibodies, with the help of non-specific immunity and the help of complement system, the natural killer cells will lyse the cell. So viral mediated natural killer cell mediated lysis of the cell happens. So this is the few things which happened for few viruses which lies their infected cell. And the most prominent examples are herpes virus, poliovirus, paramyxovirus, herpes viruses, retroviruses. So these are the examples. Now coming to the viral infection where no cell death happens, what happens there? So first of all, with your common sense you can say that they reside within the cell, they multiplicate within the cell, they propagate themselves to a certain number or to any number, and then they spread from one cell to another by not harming the parent cell. That is, steady state infection happens without cellular injury. Now persistent viral infection, what happens? One is latent infection with periodic exacerbations seen with the family of herpes viruses. So here you can see this herpes virus family I have given you in both cell lysis or cellular lysis technique and non-lytic cellular infections. So herpes family viruses can go for both the pathways, whether it can lyse the cell or it can hide within the cell for longer periods, and then someday it will come again, it will form, it will propagate and make progenies and infect other cells. Again it will go for sleep, or you can say hiding within the cell. So that is a latent infection or chronic infection, you can say periodic exacerbations. From time to time it happens to come up, wake up and again infect. Like this they stay in the body. Another one is in the persistent viral infection is the oncogenic viruses. As the name suggests, oncogenic means it forms one kind of cancer. Now that depends on the oncogenic virus type. So here hepatitis B is an oncogenic type virus. Not all hepatitis B infection leads to cancer, but it is capable of forming the cancer or inducing the cancer in the body. Then Epstein-Barr virus, human papilloma viruses, induces host cell transformation. This transformation leads to cancerous growth, okay, depending upon their target tissues. And these things are persistent viral infection. They do not actually destroy the cell, they transform the cell from one morphology to another morphology and according to the cancerous pattern. So that is the pathogenesis things. Now coming to the next part, the morphological changes. So due to this pathogenesis, a cell must have a morphological pattern change. Their surface structures, their cellular formation gets changed. Now certain viruses induce characteristic change in the host like inclusion bodies. Now what are inclusion bodies? In bacteria also you have read inclusion bodies. So here it is different. Inclusion bodies are some kind of protein or glycoprotein mixtures or lipid bodies which are coagulated or which are formed due to biochemical pathways, and it will be aggregated within the cell cytoplasm, and that can be stained by histopathological stainings, and that can be seen under the microscope. So this type of inclusion bodies, this happens due to the viral infection. The cell does not contain it in natural environment, but it forms due to the viral infection. Okay. Now eukaryotic viruses can cause microscopic or macroscopic degenerative changes. So microscopic, you know that with the help of microscope you have to see. Macroscopic means with naked eyes you can, or unaided eyes you can see the changes or abnormalities or plaques. So plaques are the clear zones where there will be no cell due to the viral infection. So this, I will show you the picture of a plaque. Okay. In host cells and tissue cells that are distinct from lysis, and these are called cytopathic effects. Now as the definition suggests, the changes which forms in the plaques, in the plaque form in the host cells and tissues, it is termed as cytopathic effect or cytologically pathogenic effects. Now these cytopathic effects, again varieties are there, differences are there depending upon the viral working process or mechanism. I have given some examples. If you can, you just remember it, like rapid crenation. Crenation is the term that means, have you seen a dry leaf? The edges of the dry leaf become sharp edges. It's like very rough, and some kind of peculiar structure, that is called crenation. That happens to the cell when it infects, in case of enteroviruses. Okay, crenation and degeneration due to enterovirus. Now syncytium formation, I have told you, syncytium is the multinucleated giant cells. So this can cause by measles, RSV, HSV, hepatitis, herpes simplex viruses, respiratory syncytial viruses and measles. Diffuse rounding and ballooning of the cell line due to HSV again, and cytoplasmic vacuolations due to SV40, simian vacuolating virus or simian virus 40, right. Then large granular clumps resembling branches of grapes like adenoviruses. So you can see here, though I have told you abnormalities and plaque formation, not all are kind of plaque formation, not all pathogenic forms are plaque formation, but there are morphological changes, there are degenerative changes which are not actually happen to a cell in a normal condition. That is classified as viral cytopathic effects and depends on several other viruses, from one virus to another virus. Now coming to virus cultivation. Okay, so if you need to isolate a virus, from a suspension, from a sample, you need to cultivate it, and you have to know how to cultivate it or how to isolate it. Okay, so again the cultivation process is different from one virus to another virus depending upon their preferred tissue. In case of bacteria, you have seen that some bacteria prefer some special media, they need some special things, or they love to grow in some media other than a basic media. So same happens in case of viruses. Okay, so viruses are generally isolated from VTMs, that is viral transport medium, and that are collected from different tissues. It may be nasopharyngeal, oropharyngeal, or it can be bronchial, whatever you see. Depending upon the viral infection site, you have to collect the sample. It can be a pus, anything, it can be a stomach fluid, it can be anything, right. So viruses are generally unable to reproduce outside that, all, that they are obligate parasite, right. They are obligate parasite, cannot be cultured in the same way as cellular organ, microorganism, like in case of bacteria, they cannot be cultured like that. They have to have a particular host cell to grow them. You cannot cultivate it outside the host cell, okay, like bacteria. So bacteriophages or bacterial viruses, the viruses are animal viruses, plant viruses, bacterial viruses. So bacteriophage is a bacterial viruses because they only infect bacteria. Now they are cultured with cells on a solid medium and lysis of the host cell occurs. So here I have seen a representative picture. These small clear zones are nothing but plaques. In the previous slide I have told you about plaques. So it is occurring in a media plate. This is a media with bacterial lawn and bacteriophages are given, and it will show you some kind of plaques, and these clear zones are nothing but there are no bacterial cells. These hazy zones or those cloudy zones are full of bacteria, okay, bacterial lawn you can say, and in these lawns these clear zones are without or devoid of any kind of bacteria. That is called plaques, okay, and this kind of plaques are also seen in case of tissues or cells in humans or animals, okay, due to viral infection, animal virus infection. Now again coming to culture, animal viruses are often cultivated by inoculating suitable host cells from embryonated eggs. So not the previous one, this is embryonated eggs, okay, fertilized chicken eggs incubated about 6 to 8 days after laying. So immediately if you culture them, you will not get the result. So after 6 to 8 days of laying, you have to inoculate the virus. I have given the picture which virus, where it should be inoculated, because again all the viruses have their own preferred tissues, their own preferred materials or zones where they infect. That is why when you want to isolate a virus from a sample, you have to inoculate it or you have to culture it in a preferred zone or preferred tissue where they want to grow. Okay, so that is the thing. Now plant viruses are cultivated in a variety of ways. Sorry, I was talking about animal. Now this is the typical picture of a embryonated egg inoculation. So here I have marked the different zones in a embryonated egg. Okay, albumin, cell, amniotic cavity, chorioallantoic membrane, inoculation, amniotic, everything I have marked. You just pause the slide and see which viruses are going where. Like this herpes simplex virus, pox virus and Rous sarcoma can be inoculated in chorioallantoic membrane. There they prefer to grow, you will get the virus in propagated form. Again, this amniotic inoculation is required for influenza virus and mumps virus. Then again, herpes simplex virus can be inoculated both in yolk sac as well as chorioallantoic membrane. In the allantoic inoculation, influenza virus, mumps virus, Newcastle disease virus and avian adenoviruses. So you can see the preferredness of these bacteria from one zone to another depending upon the embryonated egg, different zones. So this will give you a perfect isolated virus, a perfect zones of the virus where the virus can grow. Now coming to the plant viruses, they are cultivated in variety ways like cultures, plant tissue cultures, cultures of separated cells, cultures of protoplast. Protoplast, you know, the cell which is lacking a cell wall, that is called, means it have a cell membrane and cytoplasm and other organelles, these are called protoplast. So this kind of sections are used, but mainly in this study we are talking about animal viruses, okay, and to some extent bacteriophages and bacterial viruses. So all my slides, all my parts from part 1, part 2 and part 3, these all parts mainly cover the bacterial viruses and animal viruses. Plant viruses I have given you an overall picture in a short form. Now plant viruses can also be grown in whole plant. How you can grow in a whole plant? You have to damage the tissues, or you have to damage the parts of the plant and then inoculate it by rubbing with the mixture of viruses or any kind of particular viruses with an abrasion. Means you have to make a wound in the plant body and then you have to rub the virus to get infected the whole plant, or to culture them in the whole plant, the plant viruses. So going to the next, I have given again a representative picture how animal inoculation done. In the previous slide I have shown you the egg inoculation, embryonated egg inoculation and the different zones in an egg. Here I have shown you animal inoculation and primary and secondary cell culture, continuous cultures, because again when you are cultivating the animal viruses outside the animal body, because we cannot use live human beings or live animals always, it is there are ethical issues. So that is why in some time we grow them in primary and secondary cell cultures, which are not infinite. They have definitely life passages of 50, 100 or 5 or 10 passages you can give them in the outside environment in the in vitro culture to grow the bacteria, to propagate to some extent, and then you get the virus and the cell culture will be destroyed. In case of continuous or immortal cells, these cells grow from one passage to means continuous, as the name suggests, it can be many passages, okay, unlimited passages. Then there you can culture some viruses, not all viruses. Again there are preferences which cell culture is required for which viruses. Okay, now here in animal, some viruses we grow them in live animals, not humans. Here a typical picture of an animal mouse, where the sites where it can be injected is tail vein injection, subcutaneous injection, intracranial injection. So metastatic tumor growth in the lungs. So it shows very harsh to see, but actually it happens in different scientific labs and different times requirements where you need to isolate the virus and you have to use a animal. So here this picture is the passages or the mediums where the cells are kept, animal cells are kept to cultivate the viruses. Now after the cultivation you need to know the amount of virus in a sample or viral enumeration. So it can be a direct, it can be indirect method, different methods can be applied. So in one way, in a direct method, one direct approach is to count the virus through electron microscope or use of microscopy. Directly you can count the number of virus that is particularly to a cell and then apply formula to cultivate in the whole or to count the whole cellular processes or per ml or PFU. I have shown you in the previous classes. Another direct counting method is AP fluorescence microscopy. Here the virions with the are stained with fluorescent dyes to for microscopic examination. Okay, so AP fluorescence microscopy is more convenient if you see a fluorescent virus in a particular environment. Now finally, indirectly you can use directly you can use quantitative polymerase chain reaction, where the viral number, the number of the virus, not the direct virus you can see there, but the viral genome, either it is a DNA virus or RNA virus, copy numbers in a viral sample you can say with the help of quantitative polymerase chain reaction. This is the quantification of the viral number in a particular sample. Now what are the indirect methods? Indirect methods are the heme agglutination assay, plaque assay. Okay, and these are two terms you need to know, I will tell you. So mainly heme agglutination assay and plaque assay. So some viruses has the capability to bind to hemoglobin or RBCs. So when the virus bound to them, it forms a agglutination. So if normally in a suspension, suppose a saline suspension, if you give a 1 drop of blood, it will just settle into the bottom of the sample. But a particular concentration of a virus, if you add in the suspension, then that virions will join the red blood cells together and then they agglutinate, forming a network that keeps the red blood cells in suspension. So it will be a reddish suspension because the virus is agglutinated with the RBC. I will show you the picture in the next slide. And the plaque assay I have already talked about in my virus part 2. If you have not seen my part 2 of the virology decoded series, then you please go to there. I have given the formula through which MOI multiplicity by infection is calculated, as well as the detailed view of PFU and CFUs, plaque forming units, where you can see or calculate the number of plaques in a sample during culture. Now coming to when biological effects are not readily quantified by plaque and heme agglutination, because all virus cannot be formulated in these two methods or direct methods, there are highly pathogenic viruses are there, then the amount of virions required to cause the disease or infectious disease or death is curated through two terms. The lethal dose LD50 is a dilution that contains concentration of the virion dose that can destroy 50% of the host cells. So understand this like this. Suppose hepatitis B, 10 number of hepatitis B virion, suppose, listen carefully, 10 number of hepatitis B virion is able to kill 50 human beings. Okay, so this is the LD50, means if you take 100 human beings as an experiment, then it can kill 50 of those infected human beings if you use 10 number of hepatitis B virus or virion. So this is the LD50, lethal dose 50, because it is killing the animal or human. So 10 number of hepatitis B is the lethal dose 50. Okay, that is enough to destroy 50% of the host cells or organisms. In a similar sense, infectious dose 50 or ID50 means you have to be infected. How will you know you are infected? You will have symptoms. The virus will be present in your body. When it is if diagnosis happens, the virus should come positive. So the infectious dose again is ID50 is same again, but here not killing your host cells but infecting. So suppose that hepatitis B again, 5 number of hepatitis B virion is able to infect 50% of the human population or from the 100, 50 people will be infected with 5 number of hepatitis B variant, that is ID50, infectious dose 50 to the population, whatever you are using for the study. So that is the thing. Now coming to diagnosis, laboratory diagnosis can be done by isolation of viruses. First you have to isolate the viruses and then you go for diagnosis. That can be happen. That is animal inoculation, I have already shown you. Embryonated eggs inoculation, tissue cultures, organ cultures, explant cultures, cell lines, I have shown you everything. Then direct demonstration of the viruses, electron microscopy, immunoelectron microscopy, fluorescent microscopy, light microscopy for some cases, histopathological staining to demonstrate the inclusion bodies or the cellular cytopathic effects, detection of viral antigens through ELISA, direct immunofluorescence, ICTs, immunochromatography techniques, and flow through assays. Now detection of specific antibodies, conventional techniques such as HAI, neutralization test, and CFT. Newer diagnostic formats such as ELISA, ICT, again this is given in both the scenarios, but mostly ELISA, immunofluorescence, heme agglutination assay, and flow through assays are very important. Now if you are diagnosing a virus through nucleic acid probe, then nucleic acid hybridization, PCR, general PCR or traditional PCR through DNA detection by amplification, real-time PCR for DNA quantification, and real-time RT-PCR for RNA quantification, because the RNA needs to be converted to DNA for quantification. Okay. Here the heme agglutination picture that I have told you. Here you can see the normal RBC in a suspension will be seen like this. In the tube it will form a dot in the bottom of the tube suspension, but if you add some viruses in the proper quantity, it will join with the RBCs and form a heme agglutination suspension where the blood or the sample will be reddish. You will see the action. Now coming to the treatment, generally viruses do not have, all viruses do not have proper treatment, or mostly the viral infections do not have proper treatment, but you can use interferons in severe cases. Mostly interferon alpha and interferon beta are used for treatment. Interferon alphas are isolated from macrophages. Our fibroblast cells can give you the interferon beta. These are from interferon gamma does not have antiviral action, do remember that, and it can be produced by T helper cells, which are component of CMI, means cell mediated infections. So T helper cells actually induce the more production of macrophages, which will again secrete this IFN alpha and more fibroblast cells. So both are activated by T helper cells. Actually T helper cells are not producing the interferons, but it is inducing the other cells to produce the interferons. Okay, so that is why T helper cells or cell mediated immunity are mainly responsible for this interferon mediated response. So interferons have no direct action on viruses, but they induce host cell to produce certain proteins such as to turn inhibit host cell protein synthesis. So inhibit, like antibody which can directly bind to the antigen, that either may be a bacteria or a virus, interferons, they do not bind to the virus directly. They, in lieu of that, or instead of that, they give a signal to the host cell where the virus is infecting or infected, and channelize some functions within the cell, either maybe a protein synthesis inhibition or a DNA synthesis inhibition, so that the virus cannot escape that cell, or it could not infect other cells or the nearby cells. So destroy the cells within the apoptotic pathway, and that is given by the interferon signals, either maybe alpha or beta, but these are induced by T helper cells or cell mediated immunity. So the IFNs are part of a innate immunity. It can happen within hours, and it does not have any memory like B cell and T cell. Okay, so we know that antibody memory is there in our cells after a viral or bacterial infection or fungal or algal infection, but in case of virus, or sorry, in case of interferons, there are no immunological memory. These are part of non-specific immunity. And some viruses several agents including certain RNA viruses can induce IFN synthesis like Toga virus, vesicular stomatitis virus, Sendai virus, Newcastle disease virus, bacterial endotoxins, synthetic polymers, etc. These are the inducers for interferons. So, these virus or bacterial agents can directly induce the, but all are cell mediated. We have to have a T helper cell activation to induce more production of interferon alpha, beta and gamma, where interferon gamma is mostly not helpful in case of viral infections. Here I have given some antiviral drugs which are used for, as I have said, not all viruses have the treatment, or this kind of treatments are given in case of extreme cases or from chronic cases or persistent infection or life threatening conditions, but actually viral specific treatment levels are very limited. So you can pause the slide and you can memorize it, you can write it down, whatever you want. Okay. Now going to immunoprophylaxis. So this is the most efficient method to avoid viral infection. Again, not all viruses, not against all viruses we have vaccines. Actually, immunoprophylaxis is nothing but vaccines. Means you are getting pre-protection from the viral infection with a mock viral infection. Now, this kind of immunoprophylaxis can be given in three forms: killed viral vaccines, subunit vaccines, live vaccines. Now, some other methods also there like RNA vaccines, mRNA vaccines, and there are some scientific methods which are still in undergoing, but I am giving you the most popular methods here which are used, or most popular vaccines which are using this kind of mechanisms like killed viral vaccines, subunit vaccines and live vaccines. So, as the name suggests, they are inactivating viruses with heat, phenol, formalin or beta-propiolactone. They are more stable and are considered safe when given immunodeficient patients and pregnancy cases. So, they are mostly safe. Okay, killed vaccines are mostly safe, but may have some reactogenicity or adverse effects. That also depends on people to people because the immunology is different from one person to another. So, the reaction is different from one person to another. Now, coming to subunit vaccines means only a particular antigen of the virus is incorporated. Here in case of killed viral vaccines, whole of the viruses or whole cell viruses are being deactivated with chemical or physical methods, and then it is being used as a vaccine. But in case of subunit, the particular part of the virus is used as an antigen and to induce the immunity within the person where you are giving. So, this kind of example is hepatitis V vaccine. Okay. Now, coming to live vaccines, again as the name suggests, the virus is live but attenuated. Attenuated means it is weakened. It is weakened by some of the mechanisms or in vitro culture facility where some of the chemicals are used to make them weak, because they are live but they cannot induce the actual disease within the person where the vaccines are given. So, this is also a very successful method, and there are several examples. You know that polio vaccines are oral vaccines. I have given you the chart again. I will give you in the next slide the chart where different vaccines are described, and most popular vaccines are described, from where they are isolated, how they are cultivated and to which virus they are actually, or for which pathogen they are used. Now, post exposure prophylaxis, it is very important for those who are health workers or who are activating or who are doing work with patients and patient blood samples or any kind of samples. So, some kind of exposures after means you do not have the vaccine for, you have not taken any vaccine for the pathogen, but you are somehow infected or exposed to the particular virus by indirect methods with the sample, if you touch the samples or somehow your skin get pricked during sample collection. That time for immediate measures, post exposure prophylaxis or PEP methods are coming. Okay. So, again the most efficient method will be the passive immunization. For some virus, again I am telling, not all virus are having that. So, passive immunization, that means ready-made immune molecules or immunoglobulins or antibodies are given to the patient who is pricked or who are exposed for some reason during handling of patient for immediate control of the viruses, so that it cannot infect it, or in the initial phase of this or within the incubation period it get destroyed within the host cell. So this post exposure prophylaxis is very important for healthcare workers. Now here I have given the vaccine chart, most popular vaccines which are given nowadays, from where they are isolated and from where they are derived and for which disease they are activating. So not all viruses are, you are not able to remember that. Mostly you can have some idea about the which viruses are coming from inactivated, coming from subunit, and then live inactivated and live attenuated vaccines. Okay, so you can pause the slide and read it properly. So I hope this class will be helpful for you in your exams and other competitive exams. And if you have any doubt, please ask me in the comment section. You can mail me. You can join my Telegram channel. If you really like my classes, give a big thumbs up and don't forget to subscribe my channel Bioaffairs. This type of subscription and likes give me the enthusiasm to prepare more futuristic classes for you guys.